All right. Hello, everyone. My name's Ben Emerald. I'm the marketing manager at Medicinal Genomics, and I'll be your moderator for today. First, thank you for joining us to discuss the facts and myths around quantitative enumeration of yeast and mold on cannabis. There's a lot of confusion and misinformation on the topic that we hear from customers and our customers' customers, regulators, really everyone. So we thought the best way to address it would be to lay it all out in the webinar. Having said that, we're going to cover a lot of ground, and if you're not as familiar with microbiology or molecular methods like qPCR, it may seem like a lot. So we invite you to submit questions throughout the presentation, and we'll do our best here to address them either in the chat or in the Q&A session following the presentation. To do that, click the Q&A icon on the bottom of your screen. This will open up a chat window where you can submit your question. And if we don't have time to get to your question, we'll follow up via email. And of course, if you think of a question after the webinar, you can email us at info@medicinalgenomics.com, and we can respond to you that way. Also, this webinar is being recorded, and it will be available on our website later this week. So if you need to jump off, don't worry. We'll email each of you when the recording is available. With that, I'd like to introduce today's presenter, CSO and founder of Medicinal Genomics, Kevin McKernan. Kevin is a pioneer in the genomics of cannabis and hemp, dedicated to building a stronger scientific environment for the study of cannabis-based therapeutics, product safety, and blockchain technologies for tracking and verifying cannabis genetics. So take it away, Kevin.
Thanks, Ben. So we're going to cover some ground here today. I think it's important in doing so, we lay out some conflicts. We're obviously a manufacturer in the field. We make PCR kits. So you're going to see a pretty PCR heavy presentation here. I think there's some other conflicts that are worth also putting on the table. There are certainly many different business interests in testing cannabis. We have many of the growers who are very interested in getting reproducible testing from labs. And they want there to be a fairly open and free market there so they can trust any lab and get the same answer. There's also been concern that lab shopping can influence the marketplace, and that growers may be interested perhaps in lower fail rates so they can get more volume through, and sometimes those financial interests may not always be in line with the patient interests. I think from our experience working with a lot of the testing labs, they're most interested in reproducibility, in accuracy, and in scalability, which are three topics that I think are going to fit well with the PCR theme that we speak about today. And then there are regulators who are interested in addressing many of these conflicts. We all have business motivations, and their interest is to look out for the patient who may not be reflected in some of the conflicts that are here. So with that backdrop, I want to encourage everyone who's listening to this to take a real firm look at how the language of regulation is being written. And it's very different in each jurisdiction. But I think the one confounding term that is haunting the entire field and holding us back is this reliance on a colony forming unit. Because it's not a very specific term, and it's a term you can drive a truck through. And as a result, this is going to induce a tremendous amount of lab shopping and arguments over who's right and who's wrong. When you boil this down to a DNA level, a lot of those problems disappear. And now that I've made such an outrageous statement, I should back it up a little bit with some of our history in this field. And let you know that we're not quacks. We've actually been doing a lot of DNA sequencing and managing of E. coli and bacteria cultures for a very long time. I've been in the field for over 25 years, and I helped Eric Lander construct this sequencing pipeline in the Human Genome Project. I managed his research and development group there, and we built this pipeline to manage and culture 10 or 20 million different E. coli cultures in 10 months. And this was used to sequence the DNA out of those E. coli and do it quite effectively. So we became very aware of how to quantitate these organisms, how to lyse open these organisms, and how to read DNA out of these organisms in a manner which is very reflective of their reproductive capacity. A lot of this technology was then distilled into a startup company called Agincourt Biosciences, which became the largest commercial sequencing vendor in the world back in 2000 to 2005. We also developed these DNA purification tools, which you're seeing at this lower picture of these beads down in the lower right-hand corner of the slide. This is one of the more popular DNA purification systems now in the marketplace. I've seen reports that suggest that up to 80% of the market of next-generation sequencing is running off of something known as Ampure, which is a DNA purification system that we developed there at Agincourt. Beckman Coulter acquired that company. They didn't know what to do with one of our skunk work projects, so they decided to spin that out and fund it with $6 million. We had another $6 million from NHGRI, and we built a DNA sequencer that is showcased here on Science, the cover of Science Translational Medicine. This is a solid sequencer. ABI came to acquire that technology. They ended up commercializing it and capturing 35% of the market share by 2008 next to Illumina. And this was, the company was eventually acquired by Life Technologies, largely justified on incorporating the solid sequencer into Life Tech's portfolio. This system utilized massively parallel PCR of single molecules. So you had to be very cautious when amplifying single molecules billions and billions of times, such that your primers didn't misreact. And that's actually going to play a role in some of the story we speak about today. After this, this system was getting utilized for cancer patients. They were sequencing tumors, trying to find the druggable mutations in those tumors. The only challenge is we didn't have very non-toxic drugs at the time. So I got very interested in cannabis because this is a plant that made a tremendous number of compounds that were showing promise in cancer, and they're very non-toxic. So we sequenced the first cannabis genome back in 2011 and put it public. John Page followed up on this, actually, referenced some of this work and did a much more thorough job and published a lot of RNA sequencing and multiple genomes at the same time. That work came public shortly after and has been a real boon to the entire field. Now that we have multiple genomes that are done, we can predict what we can do with PCR in a cannabis background. So over the course of these years, I've luckily run into over 41,000 citations in the work that we've done. The work that I was mentored by Eric Lander, he's now the scientific advisor for President Biden. His citations are tenfold higher, one of the most highly cited scientists in the world. So we have a good understanding of what's going on when it comes to PCR. But one thing that was really strong in Dr. Lander's motivation was keeping data as public as possible because it floats many more boats and it really helps buoy the entire marketplace. And we've been doing that with cannabis. We have sequenced the largest set of cannabis DNA sequence that's out there in the world is public under our name. We've been actively putting these genomes public so that there's a resource for other people to build tools, and so the market grows faster than we can simply harvest these genomes ourselves. We've also put some very relevant publications forward, peer-reviewed, that are relevant to this very conversation. The first cannabis microbiomes were sequenced by us back in 2016. We've also discovered a couple nasty microbes that grow some really interesting mycotoxins that can be found in cannabis in these publications. And we also put public our primers for doing this back in 2016. This is a really important point because we stumbled on this for a few years when we were trying to get PCR to behave in cannabis and distinguish yeast and molds from cannabis. And when we didn't have a cannabis reference genome, we were making all types of mistakes. The primers kept reacting on cannabis, and we couldn't distinguish fungi from cannabis. We solved that problem back in 2016, and I want to demonstrate what that can do for the field. In addition to our experience in human genomics, we've put a lot of fungal genomes public. And the reason I'm emphasizing this is many of these genomes, at least 57 of them, we have sequenced without using culture. We just were able to get genome sequencing off of spores alone. This is something that can be done now, and I think it's a really important thing to underscore because we do have some regulators in this field that want to move the entire industry back to plating because they believe that's the only way you can get DNA sequencing, and that's just outdated information. You will also notice this in the SARS epidemic. You can get sequencing off of viruses without even culturing them. So we're committed to putting a lot of these genomes public, and particularly we have been putting genomes public, many of which are picked off of the backdrop here, that are fungal genomes that exist on cannabis. So we get a better understanding of the microbiome, and we have a better repertoire of what databases we need to navigate this problem. Okay, so the outline of the talk today. Now we have that backdrop. There's a couple drawbacks of both platforms, and we should be honest about them. There are drawbacks of culture, and that the vast majority of microbes are not culturable. This makes it very difficult to have a total yeast and mold test because the vast majority of your yeast and mold, in fact, won't culture. There is poor selectivity with culture, in that the conditions you need to grow yeast and mold often grow bacteria. And if you ever want to use antibiotics to get rid of the bacteria, you're often inhibiting the mold. We'll show you some data on that in a few slides forward. Plating is highly variable. It's more so variable in yeast and mold than it is on bacteria. And we'll show you why. That has to do with the fact that the mold tends to be endophytic in cannabis. It grows inside the plant and is vascularized in the plant, and you can't get that out of the plant without lysing open cell walls. And fungi have cell walls and cell membranes. So you're typically lysing open both when you want to get access to the endophytes. Fungi also have unique biological anatomy. They have conidia. We call these things spore grenades. They have a host of maybe 100 or 1,000 spores on the end of a biological feature that you can easily pipette up in one go and get very stochastic answers. You know, thousands of CFUs in one plating, and when you do it again off the same matrix, you end up with nothing. This is what is known as heterogeneous macrocolonies in the filamentous fungi world. There are also inhibitors that influence culture. When you are putting matrix into a culture system, you are not purifying any DNA out of that system. You are putting that onto a plate with all of the inhibitors present. And this is a very serious issue with cannabis because cannabis is 20% by weight made of antibiotics. It's made of cannabinoids and made of terpenes. And those antibiotics vary tremendously strain to strain. You can have 100% THC lines, things that I shouldn't say 100%, but 20% THC lines with a small fraction of maybe a 20-to-1 CBD ratio. You can have CBD dominant lines. You can have CBG dominant lines. You can have cannabinoid null lines. There's a whole variety of different cannabinoids that can be expressed, and the terpene profiles are just as complicated, and those are known to play a role in as antibiotics. So on the flip side of this, we have some drawbacks to PCR. One of the things that's brought up quite a bit is we're too sensitive. We can pick up single molecules, and that's just going to fail everything. We don't see that in practice, and we'll touch on as to why we believe that is. There are ITS and 16S targets that we utilize for the total test, the total aerobic test and total yeast and mold tests. These can have variable copy number in genomes. This is actually, we believe, the benefit, not a curse. And we'll explain that in a few slides forward. But put shortly, you would rather have more than one copy of a target in genome than something that doesn't culture at all. Dealing with a 0 or 1 problem is really difficult. Dealing with a 1 or 10 problem is very easy. Live versus dead needs to be considered when you're dealing with PCR because you're amplifying DNA, and it doesn't matter whether it's inside or outside of the organism. So there are ways that in techniques that you can use to enrich for things that are live. You can perform enrichment, that's one way. You can centrifuge things, which pellets the cells and leaves the free-floating DNA in solution, or you can use nucleases. And we have been employing a variety of those techniques in the methods. One thing that is really critical moving from bacteria to fungi is you really need to understand your cell lysis. And this is something we arguably didn't fully appreciate in the last few years and have recently modified our protocols to address. There were some standards brought to market from organisms that we've never found on cannabis, Candida in particular, that are very difficult to lyse. And we had to engineer new lysis reagents to actually tackle those. Again, that was an off-target item for us because we had not seen that organism in cannabis microbiome studies. Nevertheless, it is a standard that's been evolving in the field. And so we've had to modify protocols so we can appropriately lyse those things and be concordant with them. So where is the puck going in this world? Well, eventually this will be federally legalized here in the States. So when we look at what the federal agencies are doing, they're moving toward genomic methods. There's a genome tracker method down at the FDA. This is something that's doing genomic surveillance on the food chain, and they're doing this primarily because they recognize the vast majority of microbes and risks to human health are not culturable, and we're never going to pick them up with the older methods. The other reason they're doing this is it's faster. If we had to culture everything for the SARS coronavirus problem, we'd be in much deeper trouble than we are today because the culturing just takes too long. The same thing is being voiced down at the CDC as well with the PulseNet network, where they're using DNA sequencing to capture a snapshot of what's going on in the food chain. This was also reiterated by Frank Ioannis at the 2019 AOAC conference, where he underscored the need for genomic surveillance and the need for blockchains to track these things. So we believe this is where things are going, and we believe it speaks in the direction of molecular methods. So I'm sure everyone's tired of hearing about coronavirus, but we do need to take a look at this story. What did we use for coronavirus? We did not culture things. We did initially, in that the Jafar paper here, Rita did a great job demonstrating what CT values correlated with the PFUs. These are plaque-forming units as opposed to colony-forming units. But once they had that calibration done, they never went back. They threw it out. And the main reason for throwing it out is culturing human pathogens is dangerous. And if you think the laboratory regulations are tight now, they're about to get tighter. We've all seen the news about what's going on with the potential lab leak out of Wuhan. That is going to lead to much more stringent laboratory environments. They are not going to want you replicating human pathogens in your lab simply to measure a small amount of them. If your assay requires making billions of copies of a pathogen to measure a single one of them, they're going to be pushing you toward PCR because you can replicate these things, measure them without them being infectious using genomics. So I'll reiterate, over 1 billion of these PCR assays were done in the last year without any culture. And they were still able to sequence millions of these genomes without having to culture a single one of them. Now, another one of the reasons why they did this was quite evident in the Harcourt paper. The Harcourt paper demonstrated that the cell line and the culturing conditions that you use can drastically vary the PFU count. So if you were working with 293 T cells, you had log scale lower results than if you were with Vera cells. There was, in some cases, you can see there are almost 10,000-fold differences based on the culturing conditions that you use to get your PFU counts. So once they had this calibrated on Vera cells, they moved on and went with molecular methods all the way. So we think this is going to find its way into our life in the future. We don't know exactly when federal legalization is going to happen here in the United States, but we certainly think even before that occurs, there is going to be more scrutiny applied to labs that are managing pathogenic microbes. And it's going to push the world in a direction of using more molecular-based methods as opposed to traditional methods that require replicating the pathogen to count it. So this brings us to a common critique we hear is that plating is a gold standard. And that may have been true 50 years ago. It's been used for probably 100 years now. But the reality is it was developed in a time frame when we didn't really understand what we were counting. People began plating things before we even knew what DNA was. So we're at a stage now where we can count these things more accurately. And some of the terminology that we've been using in the past needs to be modernized with what we can do today. So we did do a study in Michigan, and Michigan was concerned that they were missing pathogens using PCR. So they set up an ERV with AOAC. We ran a large study only to find out that PCR was finding more microbes than plating was. We've then put together all of this work and published this. It's still in peer review. But this was effectively sequencing the whole genome of every single colony that was in the study to figure out what was going on. I'm going to walk you through some of the design of this study. It started with cannabis flowers that were homogenized by a third-party lab, large quantities of it, and they segregated this and binned the cannabis into 3 buckets that were measured using culture and chloramphenicol. So they came up with bins of cannabis that were high, medium, and low CFU counts as measured on something using chloramphenicol, which is a selective agent to knock down bacteria, but we also know it knocks down Aspergillus and other fungi. These samples, once they were petitioned, were sent out to fourth-party labs that would then run some of these experiments on 10-gram samples in replica, five-time replication. So during this study, one interesting thing emerged is that we looked at what the 3 different culture mediums did, because many of the BAM chapters give you some latitude here. You can use PDA or DRBC. You're not locked in on one culture system, one antibiotic, and one growth time. You have latitude there, and that latitude we want to see what does it produce. We actually had trained our qPCR system on PDA, and so we were used to the correlation there. In came DRBC, which has more selection than PDA, and we got different numbers. So this is what some of the experiment looked like by the time it reached us. Of course, the colonies got larger than we liked for sequencing, but fortunately, the people who counted these colonies in the labs in Michigan pinpointed them with markers so we could tell where to pick. We picked those colonies and began sequencing those. And right out of the gate, one thing you'll notice is that there is a log scale difference in CFUs that we're seeing between the different culture mediums. So there's a 10-fold increase in the number of CFUs found when you move from DRBC with chloramphenicol to PDA with chloramphenicol, and another 10-fold increase when you go to PDA without any chloramphenicol. So here we have a 100-fold variance in what a CFU might be. So if a regulation just says CFU, we don't really know what that means, and we're never going to get molecular methods to really zero in on this, all right? We really have to get the specifications down pat, and also debate which is the right specification, because for patient safety, they're not going to like DRBC. Maybe the growers will like this because it'll pass the most amount of cannabis. But as we'll show you, some of the most dangerous pathogens are the ones that are getting omitted while using this. So we took Aspergillus, which is the only thing to really kill people in cannabis. We took many flavors of them from ATCC and started plating them as monocultures. No matrix background interfering with this, just to see, do we pick them up with PCR? Do we pick them up with PDA? And do we pick them up with DRBC? The vast majority of them did not grow in 5 days on PDA. We had to push them out to 7 days to get some of the colonies to emerge. We picked up most of them with PCR. We have some weak CTs and rhizopus that we're trying to address. PDA picked them all up. The other thing that we noticed in the ERV study that we did in Michigan was that the standard deviations for plating were all over the map. This makes it very hard to tune something that has better standard deviations than plating against it. We're now benchmarking this and being asked to break PCR to make it as bad as DRBC. This is a real step backwards in our opinion for patient safety. So here's an example of 5x replica plating from the study where you can see the standard deviations for plating are all over the place, sometimes 42% variance on the actual count. And as you know from the prior slide, there was many fewer colonies being counted on DRBC, so PCR overestimates and tends to fail more. Now, the standard deviations on the PCR side are actually much tighter. So this is something that I think labs appreciate. They want to see highly reproducible results that are accurate and that can be turned around very quickly. Plating is the opposite of all those three things. Now, interestingly enough, the state looks like it's moving forward on DRBC despite their initial public concerns that PCR was missing things. This concern was raised by one laboratory in the state that perjured themselves to the state, claiming that our test was not picking these things up when they never had purchased our complete kits. So this whole mess in Michigan started off of a lab complaining to the state with fabricated data. And now they've moved to DRBC, which is likely going to put patients at risk because of this problem here. We have Aspergillus that is public literature showing that Aspergillus is hindered by things like DRBC. The chloramphenicol antibiotics that are present on this thing reduce the growth of this. We have got evidence of it in our PCR data and sequencing as well. This is getting pushed upon all the labs in Michigan to uproot all the gear that they've purchased in the past, put in new gear, switch to something that's less sensitive, all under the banner of patient safety when it's actually going to be worse. So we've also heard this, that plating results, the plating results actually represent the microbial population on a sample because you're only looking for things that are viable. Well, this isn't really true if there's really strong selective pressure applied. It's only going to culture the organisms that can culture, and then the moment you put selective pressure on there, you end up with something different. So let's go through all the whole genome sequencing that we did. I'm not going to, the paper has all 45 genomes there. I've just picked a few of them here to give you a taste of what's going on. You can tell these ones are on DRBC based on the pink backdrop of the colony. But what you're looking at up here in the upper left is the coverage map of these genomes. You want to see a fairly horizontal line there that shows you there is coverage. In this case, it's about 80-fold sequencing coverage on all the contigs. The contigs are sorted in size from the longest contigs on the left to the shortest contigs on the right. So we get about 2,000 contigs out of these assemblies from Illumina. And when we see multiple horizontal lines, that tells us we have colonies on top of colonies or endofungal bacteria. But you can see even in this first batch of samples with DRBC, we're picking up some Pseudomonas on here. So there is bacteria that's in fact growing on this, in this setting and contributing to the colony count, which is not fungi. These pie charts on the right are how pure the sequencing is in terms of the species that they're picking up. When this pie chart becomes a little bit more fractured, like you see on this slide over here, you know that you've got a metagenome, not a single genome. And you start to see that in some of the coverage plots. You see these other contigs that are really high or low coverage that aren't fitting to the mean. That's a sign that there's more than one organism there. Whereas these two are very clearly fungi. This one here is probably a mix. And we believe that's probably evident by looking at the size of the genome that assembled. And of course, it does bring back memories of other projects we've done in the past where there have been cases of Ralstonia on cannabis. Ralstonia is a known endofungal bacteria. It grows inside of fungi at very high copy number. You can see it here in all these green dots. This is something that's really difficult to resolve with plating because it may not necessarily rear its head on the tack plate. And if it's inside the fungi, you're really not going to know what's going on inside the fungi. You might get a colony. You might get it'll confuse colony counts and create discordance between PCR methods and plating methods. All right, so that's one artifact that we've got to pay attention to. When we move on to PDA, as you remember from the previous slide up there, we had 10-fold more colonies with PDA and chloramphenicol. But we end up with about a 50-50 mix of bacteria and fungi. So if you do the math there, that means there's about 5x more fungi in PDA than there is on DRBC. but there's also bacteria you have to sift through. So this makes counting colonies a little bit more tricky. That's a colony counting problem, not a PCR problem though. So some of these colonies are clearly fungi based on their looks, but right next to them you can see some bacteria that are growing. Another colony there, that's fungi. Here you get to some colonies that look like they're merged, or they're colonies on top of colonies, and so these pie charts widen out, and you can see the coverage charts end up giving you multiple different horizontal lines here. But this is, from a sequencing standpoint, we can still ascertain the diversity here, because these microbiomes all assemble into their discrete contigs, and we can still classify a multi-colony event is a problem for counting colonies by eye. It's not a problem for sequencing genomes. We can discern exactly what's going on by sequencing everything that's present. This is PDA with no chloramphenicol, and in this case, the ratio starts to shift for the predominant number of species here are in fact bacteria, because there's no antibiotic selection. There's still fungi coming through, and occasionally those fungi have bacteria underneath them, and it makes for some messy counting. Summary of all the 45 colonies that were shotgun sequenced is listed here. When you break them down to the species level, you can get some resolution here in the different types of penicillium that are there. This one that we published in 2016 is quite prevalent. Zamir Punja often sees this up in Canada as well. This is probably something that should be on the target list for things we should be targeting and testing, because it's in fact made bacteria pull through. PDA with chloramphenicol is about a 50-50 balance of both, and PDA without it seems to bend the ratio in the other direction more toward bacteria than for fungi. And of course, if you remember there being 10-fold differences in the number of colonies between each of these, you can ascertain that there's 5x less fungi showing up on DRBC. So this is not a total yeast and mold test by any means. This is a partial yeast and mold test that's being adopted probably because it's easier to count colonies by eye. It's a human anthropomorphism, if you will. We don't like counting colonies visually when they blend together, and DRBC does a better job making them more distinct. Another way of monitoring this problem is to put all the DNA sequences through a Simpson's diversity test. This is something that monitors what is the diversity of the organisms coming through in a particular pool of data. And of course, it comes up with the most diversity coming out of PDA versus DRBC with chloramphenicol. We'll have to send you an updated version of the slide that's not cutting off this axis here, but this is PDA with chloramphenicol. This is PDA alone, and this is DRBC. But the Simpson Diversity Index are most diverse on the PDA pool. Now, the other way to look at this is to take organisms and plate them without any matrix background in confusing things. And you plate them, in this case, we did it on PDA and PDA with tartaric acid just to get chloramphenicol out of the picture to see what was happening. We get very similar results when there aren't bacteria obscuring things. This is just a pure monoculture, same results between these two plates, but a massive drop off with DRBC. Similar, if you take Aspergillus niger and put it onto chloramphenicol and no chloramphenicol PDR plates, we get similar counts, but a massive drop off when we go to DRBC. If you take Botrytis cinerea, probably the most commonly found mold on cannabis, although not necessarily considered a very harmful human pathogen, it is an allergen. This thing has massive growth on PDA, almost none on DRBC. So yes, if you want to pass more cannabis, use DRBC. The patients won't be happy, but the growers will love it. Now, there's some other issues on using plating, which is endophytes, canidia, and inhibitors all play a role disproportionately in plating when they do over molecular methods. What do I mean by that? Well, this is Aspergillus flavus, all right? The top of there is a canidia. That's a spore, what we call a spore grenade. You can pipette one of those things, and if you break it up in the pipetting process, it will turn into 100 to 1,000 different colonies. If you don't break it up in the pipetting process, it will turn into one. So this is what's known as this heterogeneous macrocolony problem, and it means sample homogenization needs to be extreme, so extreme that we would argue you should just slice everything open and measure the DNA, because if you don't do this, you're going to get very stochastic sampling, which you see here. This is taking sample from the same homogenized cannabis flower and plating it in replica many times, and what you find is eventually you get, one or two colonies, bang, you get a canidia, you get a grenade. All right. This is why there's very high variability in plating filamentous fungi. And if you just do a single plating experiment, you're really setting yourself up for lots of lab-to-lab discordance and lots of arguments. When you dilute this further, it all goes away. So you're seeing there on the right. We did document this before, and this is present in one of our preprints where we did this with Aspergillus. And Aspergillus spores can clump like this and give you these heterogeneous macrocolonies as you go through dilution on different plating systems. And it gives you very digital results, because if you don't fully treat these things with soaps and heavily homogenize them and lyse them effectively, you can't really quantitate these things unless you can figure out how to deal with declumping them. Now let's turn to inhibitors. Many people will say, oh, there are polymerase inhibitors out there. And that may be true if you're doing a simple boil prep. If you take this sampling and just simply boil it and then put that into PCR, you are exposed to the polymerase inhibitors that might be there, from the terpenes to the cannabinoids. Who knows? There's a lot of things there that could potentially inhibit polymerases. If you're performing a DNA isolation, like we are performing in our methods, all of that stuff gets an ethanol extraction and gets washed away. And you end up with highly purified DNA that doesn't present any inhibitors. However, this is very different in the plating environment. In the plating environment, you have to live with the fact that those inhibitors could be antibiotics. And in fact, with cannabis, they are. They're very potent antibiotics. This is a very interesting paper that came out recently about another plant that synthesizes cannabinoids. This is kind of new to people in the field. It's called Trema orientalis. Interestingly enough, it doesn't produce a lot of cannabinoids. It produces something on the order of 30 to 100 micrograms per ml, so maybe 100 or 1,000-fold lower than what you might get from a cannabis plant. But nevertheless, they recognized that this small amount of cannabinoids was creating inhibition. These are ATCC organisms that are inhibited in a very strain-specific manner from this plant that makes 100-fold less cannabinoids than the cannabis plant. So we believe this is happening on the cannabis plant. We've been trying to track this down and measure it. It's a very difficult experiment to do, because you really have to control the terpenes to do it right. But even if this experiment needs more replication, because we don't have the terpenes controlled for it, but we have been seeing some strain to strain on our CT to CFU curves, all right, where the CFUs come out very different. The CTs are on top of each other. We believe that might be related to the different chemotypes that are being exposed in the plating process. They're still present when you plate, but they're gone when we purify the DNA for measuring the DNA types, or at least the fungal load that's there. The other thing that comes through in the cannabis space are sometimes citrates in the candies. These are listed on 3M's product as being inhibitory to their readout chemistry. So the E. coli is there, but the readout chemistry isn't marking it, all right? So you can see the top row up here has lots of colonies, and when you add the candy, they are decreased in number. This is presumably due to citrates being affecting this. You know, citrates are quite common. Some CalMag systems have citrate in them, so you've got to. This is not just only in candy. It could be in the aqua. It could be present in some of the growing conditions. Now, I want to talk about a paper that was presented about a month ago about PCR not being a silver bullet, and it was a good paper in that very few people have published on this problem. We've put some publications out there. Zamir Punja has put some out. There's a group called Thompson that puts them out in California, looking at cannabis microbiomes. And these are, you can probably count the number of cannabis microbiome papers on your two hands. So it's a very thin field at the moment. So we very much welcome Willow jumping into this space and measuring this. However, they got about halfway through this compared to what we did in 2016. They noticed there was discordancies between CFUs and CTs, and that can be true. However, if you go one step further, you'll notice this is not as much a reflection of the discordance in PCR as it is in the fact that none of the plating systems ever agree. And if they were to step back and do this on multiple different plating formats, they would recognize the variance they're seeing is actually related to the culture side of the equation, not the PCR side of the equation. So a few things they brought up that we agree with. The ITS copy number can be a problem, but we don't believe this is a bug. We believe it's a feature, as I mentioned earlier. And if 90% of your cultures, your organisms don't culture, you have a 0 versus 1 problem. I'd rather have a 1 versus 10 problem. In this case, they brought up ITS regions or 16S regions that had 4-fold more copies in one organism than the other, and they attributed their differences to that. It turns out their differences, we believe, are due to something else, and we'll touch on that. They also were concerned about kit contamination. This is something we think is a canard. All of our customers run NTCs, no template controls, and if any of those show signal, we'd be hearing about it, particularly if they showed a signal of a CT of 21, which would fail almost every sample. That would fail, all cannabis labs around the world would be failing if that were in fact a universal problem. We actually think this relates to some of the primers that they're using, and we'll walk you through some of the pitfalls of designing primers in absence of knowledge of the cannabis genome, which I think is what happened here. they observed some overestimation of cfus to CTs, and I think this comes down to how you measure that on the plates. And let's dive into that. So these are the primers that they use. These aren't their primers per se. They pulled them out of the literature, but many of the people in the literature who designed these ITS primers did not anticipate running them with plants as the matrix in the background. And plants have ITS regions that are very similar to fungi. So if you pick the wrong primer sets, you're going to amplify both. And the copy number of the ITS regions in the cannabis genome are far higher than they are in the fungal genomes. Same thing's true with 16S sequences, which meant to target bacteria. There are mitochondria that look like bacteria inside of plants, and they are very high copy number. And so if you don't design your 16S primers correctly, you end up amplifying plant mitochondria, thinking you have bacteria. Everything here in red is a perfect homology of the primers to the cannabis genome. A lot of them have 3' homology, which means they're going to amplify. They're perfectly homologous. So we'll comb through a few of those alignments in a moment. They're also using cyber green versus TACMan. Cyber green has an intercalating dye in PCR. That means that any product you make, whether it's a primer dimer, whether it's a cannabis amplicon, or whether it's a fungal amplicon, is going to give you signal with cyber green. TACMan is far more specific in that there's a probe in there that has to match your target, and only that gives you signal. That's very critical for navigating high copy number ITS regions, because you can design probes that actually target only one of the ITS regions as opposed to all of them, and that can get you to normalize some of this ITS imbalance that's been brought up. And as we mentioned earlier, the study really didn't look at multiple forms of plating. I think that's a major weakness of it. We've put this stuff public. You're welcome to download this. We'll have this thing available for folks to download later if they don't want to remember this in crazy long link. But you can see these alignments of their primers, the cannabis genome, are full length and perfectly landing on the 3' end. These will amplify cannabis. All right, so these can't, throw these out. There's no point in working with these in the cannabis background. You need something that won't do this. We've published some that do. We'll show you what those are in a moment. But you really want to be getting primers that are very specific, and you want to get a probe in there if you can, so you can drive higher specificity. So this is the work that they show. There's actually some nice correlation here. It's just that they're offset a little bit. These are the CQs, these are the CFUs, and so this is actually really nice work. It's just they're concerned over some of these gaps here. But these gaps are between different organisms, and the assumption in this paper was that they should grow the same, and what we're going to show you is they don't. The other thing that we'd highlight is that this is suggesting just overnight culture. We don't get away with just guessing the CT. You can't guess the time frame that you grow these either, okay? You have to be very specific here because the bacteria double every 30 minutes, the Vibrio doubles every 20 minutes, the temperature is very important, precision is needed here. This would be like hiding the CT scale on any PCR paper. The time matters. But if you grow them the same time and under the same temperature on 3 different medias, you get this picture. Here's bacillus, which was mentioned in the paper. Here's staph. If you grow them in LB versus 2XYT versus 3M, here 3M gives you 3-fold more colonies than LB and 2XYT. You move to bacillus and suddenly the colonies are enormous, and you get almost no colonies on 3M, and you get almost 20-fold more on LB, or 10-fold more, 10 to 30-fold more on 2XYT. So just the failure to have a really defined CFU it puts the whole field into chaos, because depending on which one of these methods you use, no one ever agrees. All the labs point at one another, headlines end up in the news, and then all the consumers end up not having any confidence in the regulators. All right, we really got to dial in on what we're measuring here so that we don't create this public panic and that we all understand exactly what it is we're measuring. We've put all the primer alignments here public. People can download these. We realigned them actually to CS10, which is the genome that we didn't assemble. So an independent party assembled that genome. So perhaps it's less biased than taking a genome that we put public. And then finally, I want to touch on just inclusivity and exclusivity. I think this is the real key difference between the platforms. The reason why you can have a more sensitive platform like PCR and have it fail less material is because it is more specific. We don't have bacteria showing up where they shouldn't. It also has more dynamic range. You can get about 7 logs of dynamic range with PCR, and you struggle to get 3 with plating. When you start to get out toward the 3rd log in plating, you have colonies on top of colonies, and you start to undercount. When you get extraordinarily low, you can't discern plates that are negative from plates that someone just didn't pipette on. There is no internal control in plating, so when you get a blank plate, you assume that's a pass. It could just as likely be someone who forgot to plate anything. With PCR, you have that internal control that always tells you amplified something, the reaction worked as expected. Now you can measure off of that as to whether you should take the data and go forward with it. This is something that's fundamentally missing in the plating environment. Now, if you look at inclusion and exclusion criteria on many other systems, there's usually around 30 to 50 inclusion and exclusion criteria that gets done on some of these AOAC tests, right? This is the brochure from 3M. I think there's actually a typo in this brochure. They have 5 out of 19. I think it's really meant to have 15 out of 19 here. We'll give them the benefit of the doubt there, but they're getting about 79%, if you assume that's 15, grow. But that means, you know, you've got 21% of the organisms that didn't grow, and they cherry pick these organisms for their study, right? So what happens when you don't get to cherry pick them and you take something off of a cannabis matrix, right? The numbers are probably going to be much lower than this. And then down here in exclusivity, they get 21% of the colonies are bacterial. All right, so these things are not excluding the bacteria, and they're not picking up all the fungi. When you look at this with PCR, the numbers are much cleaner. The other thing that's going on with PCR is that you can go beyond, each one of these organisms from ATCC may cost you $100 to $300 to order. So if you want to order thousands of them, it's going to take you a very long time. It's going to be a very expensive endeavor. However, with PCR, you know what amplicons amplify with your primers, and then you can extrapolate how many other organisms would amplify if you just scan them against NCBI. And we can get into the thousands of organisms with PCR that will have confidence of amplification. That cannot be said with plating because you can't anticipate what's going to plate based on a given carbon source. It's very difficult. So when we look at this stuff with PCR, the selectivity is much better. We have 96 out of, we basically ordered 52 organisms from ATCC. 50 of them we're able to detect in 90 minutes, whereas you're looking at what's going on in plating, and it's like 15 out of 19 in 3 days. Much rather have 90 minutes and 96% than 79% in 3 days. Then you look at the bacteria side of things, they're getting, 21% of the organisms that are being bacteria. We don't get any. All right. This is why other systems this is why culture is failing more, is because when bacteria is present, and some people do use bacteria as some type of additive for beneficial microbial usage in the plants, that stuff will carry through and can wreck your plating counts. All right. The other thing you can do is take these primer sequences. These were published. You're welcome to grab them and check yourself. You can take these things and scan them against well-curated databases. The important thing to do is to make sure they're PacBio assemblies, because the ITS regions don't always assemble perfectly with Illumina data. But if you curate the data appropriately, you can see that we can reach thousands of organisms. I've not seen any total yeast and mold test out there that can claim that they've grown thousands of organisms and prove that they all grow on their culture system. They usually stop at around 100 because it's too expensive and takes too much time. Not to mention a lot of them just don't grow. So this kind of brings us to the final point, is does qPCR fail more samples? Well, in the case of Michigan, we certainly did, because they moved to a system that has very high selective pressure. However, when you look back at some of the publications, we never resorted to a single medium. When we do this in the past, we would triangulate this across 3 different culture systems. You see down below 3M Petri films, BioLumex, and SimPlates, and we would have to average the CFU results from those 3 platforms and then calibrate the CFU curve according to the average of the 3 platforms. Now that means when you compare yourself against any given one of them, you're not going to be perfect. But it does mean when we're dealing with a marketplace where every customer might be comparing this to a different plating system, it's probably the safest approach. But what else you will notice going on in this study is we looked at what the DNA content looked like directly off the cannabis plant and what happened after you cultured it. And what we found was radically different results based on the culture platform that you used. This is public. You can go view it for yourself. It's been peer reviewed. It's a transparent review system. I think John McPartland and Ethan Russo were involved in that. And so there's a wealth of information on there as to how things change when you apply culture to the actual microbial content. But the real problem here is that we have exquisite sensitivity and specificity with PCR, and culture has no idea what the hell it's measuring. So it's very difficult to have these discussions with folks on why are there differences between these platforms when the culture folks are kind of staring at tarot cards and we're bringing forward whole genome sequencing data. Now, the other thing you can do with PCR once you have this running is you can tackle things that don't culture. Powdery mildew doesn't culture. The viruses don't culture. So we have helped many grows using hops latent viroid testing and lettuce chlorosis virus testing using PCR so they can avoid problems like this that happen. This is a grow that actually adopted PCR and was able to clean out their LCD and their hops latent viroid infections and get their yields back up another 40% because this was wrecking them late in grow. So it's a platform that we don't yet know whether these viruses play any role in human health, but they certainly play a role in plant health. And so it's a universal platform that can help plant health and also bring some color on human health. All right, so in conclusions here, there is a clinical paradox going on with culture-based methods. You really need robust homogenization just for good sampling statistics and to deal with these clumps that occur with Canidia. And this is orthogonal diviability. If you want to get at the endophytes, you have to aggressively homogenize the sample, which is going to shred up microbes so they're not viable. So the best thing we believe is just lyse that all open and measure the DNA, and you're going to get the best proxy for what's going on. The number one clinical liability in cannabis is Aspergillus. Without a doubt, if you comb through the clinical literature, most of the aspergillosis that's going on and any clinical death, it's not THC content, it's not mycotoxins, it's not heavy metals. It is people that are getting aspergillosis, usually immunocompromised, and this is an endophyte. If we adopt methods that select against Aspergillus growth with DRBC and against endophytes because we're resorting to culture, we are playing a game that the patient's not going to be pleased with. So we would argue you want to move to molecular methods that can properly lyse all these things, and that you want to get the regulations to be adequately allow those things, if you will. If you anchor everything in CFU language, and particularly very vague and non-specific CFU language, you're just gonna invite laboratory fights for the next decade. Now, in doing so, you have to pay attention to live dead, and we have, that's probably a whole other webinar is how we deal with that, but live dead can be tackled with PCR. That is another myth that it can't handle that, but there are multiple techniques that are being utilized in the field to manage the live dead situation, from centrifugation to enrichment to using nucleases, but it is doable. So the other infections that we have seen in the field, there has been Fusarium cutaneous infections. All right, this is also a plant endophyte. There have been cryptococcal meningitis infections from pre-rolls. This was measured in Bakersfield, California. This is also an endophyte. The other things that have come up are Salmonella and Mycobacteria. These are epiphytes. These are bacteria that are on the surface. All right, these probably can be addressed with either PCR or plating. Now, it's really the epiphytes that we have to turn our attention to. Now, there is a lot of jurisdictional heterogeneity out there. Not everyone's requiring total yeast and mold. California luckily got rid of it, and they moved toward all molecular methods looking at Aspergillus in a species-specific manner. There's also this question of what is the clinical actionability of 10,000 CFUs or 100,000 CFUs of these total things, which we've shown you are not total things, they are partial things. And they're not necessarily measuring anything of clinical relevance. They're just counting colonies at this point, and they don't even have a good sense of what the colony is, whether it's bacterial or fungi, or whether it's harmful or not. And we're doing this exercise all well knowing that the vast majority of microbes don't even culture. So it's really an interesting, I think, security theater. Now, the other thing that comes into play here is when you start using these very grotesque tools like total yeast and mold and total aerobic count, is you basically eliminate the grower's ability to use beneficial microbes, because those light those systems up. I don't know where that pushes them. Does it push them into pesticide use? Where do they go? But they're going to need to do something to fight back pathogens, and putting in these non-specific tests with no clinical utility basically strips away one of their most green and environmental tools that they could be using. So conclusions, we think the CFU is an abused non-specific term. Even in the BAM, there is a lot of latitude on what type of plating you can use. And this means people could adopt and lab shop based on labs who use the most stringent plating systems that fail the least amount of material. We think the regulation should be modernized in line with where the FDA and the CDC are going. We believe the desire to actually keep this grounded in culture, because that is because you believe that's the only way that you can sequence a genome, is patently false and can be shown throughout the literature that is no longer the case. You can sequence genomes without culture quite readily today. It's quite modern, and it's done routinely for even RNA viruses like SARS. We think the total tests are, security theater, as I've mentioned before. They're not giving us any clinical utility. They're creating a tremendous amount of friction in the labs, making labs uproot and change the different testing techniques that they need, and not necessarily giving any benefit to the consumer. In fact, we believe what happened in Michigan is going to add risk to the consumer, even though that was the exact opposite of what the regulators were looking for. So we believe that this can be tuned with PCR, but we need to get together with many of the regulators in the labs to kind of structure what those regulations should be and how they should be written. And we ask everyone in this audience to try and share this information with them if they can, because we do think it is time to recraft these and modernize these regulations. Species-specific testing would make far more sense in our mind. With that, I'm going to leave it for questions because I think I'm running out of time.
All right, thank you, Kevin, for a great presentation. Just to remind everyone, if you do have a question to ask of Kevin, the Q&A icon at the bottom of your screen is the best way to do that. We will ask those as they come in. Just give a minute to see if any populate here. All right, this one's from Sherry Pearson. How can we move forward with the state of Maryland tomorrow is moving to culturing?
Yeah, I don't know how to get through to the state of Maryland. There is nothing about what they're doing down there that can be justified in the scientific literature. They want to push everything into culture, and the justification I heard was that they want to do that so that they can sequence things. So someone down there needs to read a biology book that they can actually sequence these things without culturing. And in fact, most people, before they want to sequence something, is they purify the DNA like you do for PCR. So I suspect there's going to be a lot of turmoil in Maryland, and that this needs to get out to the patients. The patients need to know that if they push them in the direction that they're going, there's going to be more cannabis on the market that has risk associated with it. And that liability should fall right back to the regulator who's doing this on no scientific grounds whatsoever.
Okay. Another question here about live dead. I was wondering if you could expand a bit more on what capabilities are out there for that.
So live dead has, there's a couple ways we've skinned that cat. There is a step in our yeast and mold. There's two different ways we've come about trying to address this. Our yeast and mold system right now has an enzymatic lysis that's lysing open more fungi than we were doing last year. This is a recent update that we've put out to address the candida issues. That enzyme is not compatible with the nucleases that we were using for live dead. So we've switched that over to a centrifugation-based step where you spin and pellet the cells down, leave the free circulating DNA in solution with that pelleting step, and then isolate that, that pellet. What we used to do in the past was use a nuclease. We can still use that nuclease for live dead in our other assays, and that's what we do. We use a nuclease that doesn't penetrate cell membranes and eats up free circulating DNA. But however, right now that enzyme is not compatible with the TLP enzyme we're using to lyse open the fungi. So we have a bifurcated approach on the test right now for live dead. And that's one way to address it. The other way we've seen other people address this is to grow a little bit, enrich for X amount of time, and only the replicative organisms will replicate. The ones that are dead will get drowned out. You then have to adjust your CTs accordingly to your doubling rate. And so that's an alternative approach, but it can be dealt with, and I think the providers in the field are all coming up with their own creative ways to do this.
All right, great. This next one is from Brett Lorenz. How much of a risk do you think mycotoxins are in the cannabis market?
It's a very good question on mycotoxins. I have not seen any papers suggesting poisoning of these things. I know that this is something that's a chronic problem. You don't tend to get an acute effect with mycotoxins. Tends to be damage over long haul. I've also not seen a lot of literature on the failure rates for mycotoxins. I know we have a lot of states that are mandating these things, but when I have surveyed them and asked around, most of the labs say they rarely ever fail something for mycotoxins. So, mycotoxins are usually synthesized in a saprophytic state, and I don't know that the cannabis plants ever get there. I think they're mostly at a state where they've been quickly cured and they get to the customer before anything rots. And so this is, a long time ago, we had a program trying to build an immuno PCR system that would pick these things up. And we put it on ice because we were just not seeing enough data in the field that the fail rates were high enough to justify the test. So we thought longer term, people may, if we have 5 years of no fails of that, I would imagine regulators will just pass on it, no longer insist on it. But the one area we have thrown up some caution is that there are a paper that we published showing that there are mycotoxins that are being made by some penicillium species that interfere with the anti-epileptic properties of CBD. And that might be something that's really meaningful for the epilepsy crowd. So that's an area that needs more work to know how much citrinum and paxilline can we actually tolerate on this when you're trying to extract these things for patients in the epilepsy field. The extraction process might enrich these things above and beyond the levels that they're in the flower, and that could have some negative consequences. But we're not scanning right now. I haven't seen any regulations out there that are looking for the penicillium-based mycotoxins. So I think that's kind of an unknown field at the moment.
Okay, another question here from Eric Lieber, who is an Arizona local and wants to know how we can find out if his state is using culture testing or PCR. Any guidance there?
I'm sorry, you broke up a little bit there.
Oh, I'm sorry. I'll ask it again. There's someone here from Arizona who is curious to find out if there's anywhere he can find out if the state is using or allowing culture or qPCR testing.
That's a good question. I think I have to deflect that to somebody else here who might be more familiar with exactly what's going on in Arizona. I did read, like this week in the news, that there was some Aspergillus outbreak in Arizona. And I also read somewhere that they're moving to plating, which didn't seem, or they're removing Aspergillus testing altogether. And that seemed odd considering they just had some news of that in the media. But yeah, I'll have to get back to you on that one. Someone here knows the answer to that. I just don't have it at the top of my head.
Yeah, Eric, if you're listening, if you want to shoot us an email, info@medicinalgenomics.com, we can route that to the appropriate person to follow up. All right, one more here, coming back to live dead. Does your solution for live dead remove only free DNA, or will it also remove DNA from dead cells that have not released their DNA into solution?
So that's a good question. The nucleus does not penetrate the cell membrane. And the question is, if there's a ruptured membrane, will it get in there and still chew up the DNA inside the cell? I don't think we have proof of that. I would hypothesize that it would, but I've got to think through how to set up an experiment that would sort that out. That seems to be a bit, that might be a bit tricky to without getting some microscopy involved. But I would, we do know the weight of the molecular weight of the actual enzyme. So it doesn't need to be a very big hole for this thing to get in. So I would imagine it would chew it up if the membrane is ruptured, but we've not set up any experiments to directly address that question.
All right, and one more came in, and I'm going to sneak it in here. Have you heard of any interference between rapid 3M yeast and mold plates and certain cannabis products, such as seeds or nut butters?
Oh, that's a good question. I have not heard anything directly on those. I've heard mostly from candies, but that is, there is some literature out there on in the food testing space that when you move to a lot of spices, you have to revalidate things because the spices have lots of active compounds that can throw off some of the testing platforms. I'll have to dig up those references. I can't recall whether they were directly addressing 3M or bioMérieux or what platform that was specific to, but I think coumarin was one that came up as being an inhibitor to many of these things.
All right, I think that's all the time we have today. Thank you, Kevin, for joining us and sharing your knowledge on this topic. And thank you everyone who joined us today. We will be sending out an email with the recording of this presentation and also a list of all the different resources that we've put together over the years of blogs and articles and papers around this topic for more information. So be on the lookout for that, and thanks again.